Good morning to everyone. I am Dr. Latha, Professor of Physiology, Sri Lakshmi Narayana Institute of Medical Sciences. Today I am going to explain about the cerebrospinal fluid. Before going into the cerebrospinal fluid, you should be familiar with the meninges, some of the basic concepts. So in that first one is the meninges of the brain. So meninges are nothing but coverings of the brain. Brain enclosed within a cranial cavity by three connective tissue layers: outer dura mater, middle arachnoid mater, and inner pia mater. So this picture showing the coverings of the brain, and this one is the dura mater, and this one is arachnoid mater, middle arachnoid mater, and this one is the inner pia mater. So first one, outer dura mater, it is composed of two layers, outer endosteal layer and inner meningeal layer. So these two layers are fused everywhere except in some places where they form fold or venous sinuses are enclosed between them. The space between the outer dura mater and the middle arachnoid mater is called subdural space. So next one is the middle arachnoid mater. It is a translucent avascular layer. It is connected to the pia mater by filamentous fibers. The arachnoid mater has minute protrusions are called arachnoid villi that passes through the fenestrae present in the dura mater and project into the venous sinuses to allow the escape of Csf into the venous sinuses. And third one is the inner pia mater. So this is closely and continuously covering the external surface of the brain and spinal cord. This pia mater is thin and highly vascular membrane. It dips into the sulci of the brain. The folds of pia mater enclose tufts of capillaries called choroid plexuses. These choroid plexuses only secretes the cerebrospinal fluid. The space between the pia mater and the middle arachnoid mater is called subarachnoid space. This space is filled with cerebrospinal fluid. So meninges, we discussed about meninges. Meninges, there are three coverings of the brain: outer dura mater, middle arachnoid mater, and inner pia mater. So outer dura mater is outer covering, and it is fused everywhere except where it forms folds and where the venous sinuses are enclosed. Okay, and arachnoid mater, it is a middle layer. It has a minute protrusions called arachnoid villi that will penetrate into the fenestrae of the dura mater and enters into the venous sinuses. And third one is the pia mater. This is the continuous covering of the external surface of the brain. It covering continuously and closely the external surface. It is thin and highly vascular. It dips into the sulci of the brain. So the folds of pia mater enclose tufts of capillaries called choroid plexuses. The space between the pia mater and the arachnoid mater is called subarachnoid space. This space is filled with cerebrospinal fluid. And next concept is cisterns. Cisterns are nothing but these are the enlarged subarachnoid space. That is, subarachnoid space is enlarged in some places to form the cisterns. There are four major cisterns and some of the minor cisterns also present. So the four major cisterns are superior cistern, cerebellomedullary cistern, Fontaine cistern, and interpeduncular cistern. Superior cistern, it is located dorsal to the midbrain. cerebellomedullary system. It is also known as cisterna magna, which is located between the cerebellum and the medulla. Pontine system, it is located near pons. Interpeduncular system, it is located at the cerebral peduncles. So here you can see the picture of systems. So first one is the superior system. So superior system, it is present dorsal to the midbrain, and next one is the cisterna magna or cerebellomedullary system. It is present between the cerebellum and the medulla. And third one is the pontine system. It is present near to the pons. And fourth one is the interpeduncular system. So it is present in the cerebral peduncles. So the minor systems are cistern of optic disc, cistern of lamina terminalis, cistern of corpus callosum, perimesencephalic systems, cistern of lateral sulcus, and lumbar system. Next one is the ventricles of the brain. There are 4 ventricles, 2 lateral ventricles, 1 third ventricle, and 4th ventricle. So ventricles are nothing but they are hollow cavities in the brain. There are 4 ventricles, as I said. There are 2 ventricles, 2 lateral ventricles, 1 third ventricle, and 1 fourth ventricle. In this picture, you can see the 2 lateral ventricles, and this one is the third ventricle, and this one is the fourth ventricle. So these 2 lateral ventricles are located symmetrically in the cerebral hemisphere. And third ventricle, it is located in the diencephalon, and fourth ventricle, it is enclosed in the pons and medulla. The 2 lateral ventricles communicate with the third ventricle by 2 openings. You can see in this picture, the 2 lateral ventricles communicate with the third ventricles by 2 openings called foramen Monro or interventricular foramen. And this third ventricle, so here in this picture you can see, this third ventricle communicate with the fourth ventricle through a narrow passage that is called aqueduct of Sylvius. This fourth ventricle narrows down caudally as it enters the spinal cord that continues as central canal in the spinal cord. The fourth ventricle connects with subarachnoid space by the foramen of Megenthi, which is the medial opening in the roof of fourth ventricle connecting to the cisterna magna. And 2 small openings, the foramen lasca, which is leading into the cisterna pontis. And each ventricles contain choroid plexuses. So there are 2 lateral ventricles, 1 third ventricle and 1 fourth ventricle. The 2 lateral ventricles communicate with the third ventricles by 2 openings called foramen Monro, and this third ventricle communicate with the fourth ventricle through a narrow passage that is called aqueduct of Sylvius, and fourth ventricles narrows down caudally. As it enters into the spinal cord, they continue as a central canal in the spinal cord. And this fourth ventricle connects with the subarachnoid space by foramen of Megenthi, which is the medial opening in the roof of fourth ventricles that connecting the cisterna magna. Cisterna magna, it is nothing but it is a cistern present between the cerebellum and the medulla. And the fourth ventricle connects to the cisterna pontis via the foramen of lasca. So each ventricle contains choroid plexuses. So next one is the choroid plexuses. Some arteries with the covering of the pia mater passes through the brain substances. to reach the ependymal layers of the lateral, third, fourth ventricles, where they break up into complex capillary network and become lined with much folded ependymal cells. The blood vessels of this lining epithelium constitute choroid plexuses. This epithelium becomes differentiated into cuboidal cells containing mitochondria, granules, and vesicles, indicating that the cells involved in active metabolic process. So this is the structure of the choroid plexus. So these are the ependymal cells, and this one is capillaries. The capillaries lined by the ependymal cells. So these choroidal plexuses mainly involved in the formation of cerebrospinal fluid. So now we come to the cerebrospinal fluid. Cerebrospinal fluid is a clear, colorless alkaline fluid, and it is having the pH of 7.31 to 7.34, and it is having the specific gravity of 1.005, and the ions are it contains ions, and the osmolarity is similar to that of the plasma. The volume of the CSF is 100 to 150 ml, out of which 20 to 25% present in the ventricles, and most of the cerebrospinal fluid present in the subarachnoid space. So this table shows the composition of the CSF and the plasma. The comparison of the ions and organic substances present in the plasma and cerebrospinal fluid. The organic substances like protein, which is 30 milligram per deciliter in plasma, but only 6 milligram per deciliter in CSF. Next one is the glucose. It is 70 to 110 milligram per deciliter in plasma and 40 to 70 milligram per deciliter in the CSF. So glucose level is less when compared to plasma. Glucose and protein levels are very less when compared to plasma. And sodium chloride. Sodium chloride it is 560 to 630 milligram per deciliter in the plasma and 720 to 750 milligram per deciliter in the cerebrospinal fluid. Cholesterol, which is 130 to 230 milligram per deciliter, but in the CSF there is no cholesterol present. Urea it is 20 to 40 milligram per deciliter, but in the CSF it is 10 to 30 milligram per deciliter. Creatinine, it is 0.6 to 1.5 milligram per deciliter, and in the CSF it is 0.5 to 1.9 milligram per deciliter. Uric acid is 2 to 8 milligram per deciliter, and in the CSF it is 0.5 to 2.5 milligram per deciliter. Phosphates phosphates are 2 to 4 milligram per deciliter and in the plasma, and in the CSF it is 1.2 to 2 milligram per deciliter. So next, inorganic ions. The inorganic ions, first one is the sodium ion. It is 136 to 145 milli equivalent per liter in the plasma, but 137 to 145 milli equivalent per liter in cerebrospinal fluid. And potassium ion is 3.5 to 5 milli equivalent per liter in the plasma and 2.7 to 3.9 milli equivalent per liter in the CSF. So the potassium concentration is very less in the CSF when compared to plasma. The calcium ion concentration is 4.2 to 5.5 milli equivalent per liter in the plasma and 2.1 to 3 milli equivalent per liter in cerebrospinal fluid. Magnesium concentration is 1.3 to 2.1 milliequivalent per liter in plasma and 2 to 2.5 milliequivalent per liter in the cerebrospinal fluid. Chloride ions concentration is 98 to 106 milliequivalent per liter in plasma and 106 to 122 milliequivalent per liter in the CSF. So chloride potassium concentration is less when compared to plasma, and chloride concentration is more when compared to plasma. Chloride and sodium chloride concentration. And bicarbonate ions 21 to 27 in plasma and 20 to 24 in cerebrospinal fluid. So the pH is 7.36 to 7.44 in the plasma, and the pH of the CSF is 7.31 to 7.34. The osmolarity of the cerebrospinal fluid is similar to that of the plasma, that is 285 to 295 milliosmol per liter. Plasma is having 290 to 295 milliosmol per liter. So these are the composition of the plasma and cerebrospinal fluid. So in the cerebrospinal fluid, glucose, protein and fat concentration is less when compared to plasma. Fat and potassium concentrations are less when compared to plasma, and chloride concentration is more when compared to chloride, and chloride concentration is more when compared to plasma, and osmolarity is almost similar to that of the plasma. So next one is the pressure of the cerebrospinal fluid, CSF pressure. So in the recumbent position, it is 120 to 180 millimeter of water. Sorry, centimeter of water, and 300 centimeter of water in sitting position due to effect of gravity. The pressure fluctuates about 10 millimeter. Okay. So millimeter, that is not centimeter. 120 to 180 millimeter of water in the recumbent position, 300 millimeter of water in the sitting position due to effect of gravity, and the pressure fluctuates about 10 millimeter of water during the respiration. So during coughing and crying, the pressure of the CSF increases. So next one is the formation of CSF. So it is, as I said already, it is mainly formed in the choroidal plexuses. So the site of formation is choroidal plexuses, which are covered by the specialized ependymal cells. The rate of formation is 0.3 ml per minute or 500 ml per day. So mechanism of formation. It is formed by combination of passive diffusion, active transport, and facilitated diffusion. So this picture showing the formation of the CSF, and this yellow color cell is the ependymal cells. Okay, inside the cell, the carbon dioxide combined with water to form the carbonic acid in the presence of carbonic by the action of carbonic anhydrase enzyme. So this carbonic acid is highly weak acid and immediately dissociate into hydrogen ion and bicarbonate ions. These bicarbonate ions diffuses into the cerebrospinal fluid, and this hydrogen ion, it enters into the blood in exchange for the sodium ion by the pump called sodium hydrogen exchanger. Then chloride ion, it is passively diffused from the blood to cell, then from the cell to CSF. The sodium ion which enters inside the cells, that are enters into the CSF in exchange for the potassium ion. Because of this, the sodium and chloride concentration is more in the CSF. So next one is the circulation of cerebrospinal fluid. Cerebrospinal fluid, these are formed in the ventricles. From the ventricles, it goes to the subarachnoid space. So it is mainly formed in the lateral ventricles. So from the lateral ventricles, it passes into the third ventricle through the interventricular foramen or what is foramen of interventricular foramen or foramen of Monroe. From the third ventricle, it goes to the fourth ventricle through the aqueduct of Sylvius, and from the fourth ventricle, it goes to the subarachnoid space of brain and spinal cord via the foramen of Latha and foramen of Megenthi. So foramen of Megenthi, it goes to the cisterns present between the cerebellum and medulla, and through the foramen of Latha, it goes to the Pontine cistern. So this cerebrospinal fluid formed by the lateral ventricles. In this picture, you can see these are formed by lateral ventricles. So after formation, these are circulated from the lateral ventricle to the third ventricles via the foramen of Monroe, and from the third ventricle it goes to the fourth ventricles via the aqueduct of Sylvius. From the fourth ventricle it goes to the subarachnoid space through the foramen of Latha and aqueduct of Sylvius. Through the foramen of Megenthi. So this picture also showing the formation and circulation of the CSF. So next one is the absorption of CSF. It is mainly absorbed via the arachnoid villi into the dural sinuses and the spinal veins. 4/5 of the absorption occurs via the cerebral arachnoid villi and rest via the spinal arachnoid villi. The hydrostatic pressure in the subarachnoid space is higher and osmotic pressure is lower because the cerebrospinal fluid contains less amount of proteins. So osmotic pressure is lower than in the dural sinuses and the spinal veins. So because of this, it facilitates the fluid movement. So next one is the functions of CSF. The CSF, it gives cushion effect to the brain along with the blood-brain barrier. So apart from this, it acts as a lymph because there is no lymphatic system in the brain. So CSF, it acts as a lymph. And second function, it provides a proper chemical environment for the optimal activity of the neurons. And third one is, it is useful for diagnosis of brain disorders as it reflects their brain function. And next one, it to protect the brain from injuries by acting as a shock absorber. So it acts as a fluid buffer. So that it acts as a fluid buffer. It protects the brain from the injuries by acting as a shock absorber. So next function is, it takes part in maintaining a constant internal volume of cranium. That is called Monroe-Kelly doctrine. And next one is the continuous formation and drainage helps in preventing accumulation of metabolic products within the brain. And next one, it provides nutrition to the nerve cells. And eighth function is control of respiration, because the carbon dioxide acts on the medullary chemoreceptors by altering the pH of CSF and brain extracellular fluid. So the carbon dioxide first dissolved in the CSF, then only it will act on the chemoreceptor. And finally, it plays very important role in the transport of hormone and hormone releasing factors. So now we'll come to the applied aspects, hydrocephalus. So first one is the hydrocephalus. So it is abnormal accumulation of cerebrospinal fluid in the cranium. So the main causes for the hydrocephalus are obstruction to the cerebrospinal fluid circulation, and second cause is excessive production of CSF, and third one is interference with absorption. So on the basis of causes, it is divided into two types. One is internal or non-communicating hydrocephalus, external or communicating hydrocephalus. The internal or non-communicating hydrocephalus, it occurs when the obstruction is within the ventricular system or roof of the fourth ventricle. Second, external or communicating hydrocephalus, this occurs due to the obstruction in the subarachnoid space or arachnoid villi. The excess fluid is mainly in the subarachnoid space in the external or communicating hydrocephalus. So this picture showing the difference between the normal ventricular system and ventricular system in the hydrocephalus condition. So you can see the increased size of the ventricles in the hydrocephalus due to accumulation of cerebrospinal fluid. So finally, collection of CSF. In two ways we can collect the CSF. One is lumbar puncture and another one is the cisternal puncture. So first one is the lumbar puncture. A special needle of 1 mm bore and 8 cm long withdrawable stylet is introduced into the subarachnoid space between the L2 and L3 or L3 and L4 vertebra. Lumbar puncture is done with the patient lying on one side with the trunk flexed and knee and chin are approximated. The local anesthetic is introduced into the overlying skin. So this is the position of the collection of the CSF by lumbar puncture. Okay, the trunk is flexed and the knee and chin should be approximated. And the CSF is collected between the L2 and L3 vertebra or L3 and L4 vertebra. Between the L3 and L4. Okay, so this picture also showing the collection of the CSF from the subarachnoid space. And next one is the cisternal puncture. So in this, the needle is introduced between the occipital bone and the atlas vertebra into the cisterna magna. So fluid is, the CSF is collected from the cisterna magna by introducing the needle between the occipital bone and atlas vertebra. So why we are collecting the lumbar CSF by puncturing the lumbar puncture or cisternal puncture? So first one is the first purpose is for diagnosis. So in bacterial infection of the meninges, the cell count with many polymorphs increases and glucose decreased. So mainly for the diagnostic purpose, we are collecting the CSF either by lumbar puncture or cisternal puncture. And second one is to measure the intracranial pressure. And third purpose is to introducing radio opaque substances for myelography to to visualize subarachnoid space in the spinal cord. And fourth is to relieve pressure. Small amount of CSF may be removed slowly to relieve intracranial pressure when it is raised. And fifth is to introduce drugs like spinal anesthesia. We are introducing the syringe, okay, into the subarachnoid space. Okay, so these are the purpose of lumbar puncture. So CSF is nothing but it is a fluid present in the cranial cavity that cushions the brain. So normal CSF is clear, colorless, alkaline fluid, and pH is 7.31 to 7.34. Specific gravity is 1.005, and the volume is 100 to 150 ml, out of which 20 to 25% present in the ventricle and remaining present in the subarachnoid space. So this table showing the composition of the CSF, composition of the organic substances and ions present in the CSF and plasma. So the protein is very less in the CSF and glucose is less. Then sodium chloride is more in the CSF and uric acid are very less when compared to plasma. Then potassium ion is less when compared to plasma. Then calcium ion also less when compared to plasma. Then chloride ion is more when compared to plasma, and the osmolarity is similar to that of the plasma. CSF pressure is 120 to 180 millimeter of water in the recumbent position, 300 millimeter of water in the sitting position, and the pressure fluctuates about 10 millimeter of water during the respiration, and during coughing and crying it increases. So formation, it is mainly formed in the choroidal plexuses by combination of passive diffusion, active transport, and facilitated diffusion. The rate of formation is 0.3 ml per minute or 500 ml per day. So this picture explains the formation of the how the ions formed and transported into the CSF. And next one is the circulation of the CSF. The CSF formed by the two lateral ventricles or passes to the third ventricle through the foramen of Monro, and from the third ventricle it goes to the fourth ventricle through the aqueduct of Sylvius, and from the fourth ventricle it goes to the subarachnoid space through the foramen of Megenthi and foramen of Lasca. So this picture also showing the circulation of the CSF. And next, absorption. So it is mainly absorbed by the arachnoid villi and into the pleural sinuses, venous sinuses, and into the spinal veins. So 4/5 of the absorption via the cerebral arachnoid villi and rest via the spinal arachnoid villi. The hydrostatic pressure in the subarachnoid space is more, so that it facilitates the fluid movement. And next, functions of CSF, it acts as a limb. It provides the proper chemical environment. It is useful for the diagnosis of brain disorders, and it helps in the absorbing the shock, and it maintains the constant internal volume of the cranium, and it prevents the accumulation of metabolic product by continuous formation and drainage, and it provides nutrition to the nerve cells. So it also involved in the control of respiration. It also involved in the transport of hormone and hormone releasing factors. So in the applied aspect, hydrocephalus. The next one is the collection of CSF. In that we discussed about lumbar puncture and cisternal puncture. The lumbar puncture, the needle is introduced between the L2 and L3 vertebra into the subarachnoid space, or needle is introduced between the L3 and L4 vertebra into the subarachnoid space, and the position of the subject is the trunk is flexed and chin and knee should be approximated. And next one is the cisternal puncture, so in which the cerebrospinal fluid is collected from the cisterna magna by introducing the needle between the atlas and occipital bone. The main purpose of the lumbar puncture and cisternal puncture are for the diagnosis, to measure the intracranial pressure, and for introducing the radio-opaque substances, and to relieve pressure, and to introduce drugs. Thank you.